Hi, welcome. Thank you so much for joining us today for the second in our series of Fast Plant webinars. So a quick introduction to our panelists today. My name is Julie Stubbs, and I am the Product Manager for Zoology, Botany, and Microbiology at Carolina Biological. I will be joined today by several guests. Bob Kuhn is a teacher with 22 years of experience. He teaches honors and AP Biology at Centennial High School in Roswell, Georgia. Claudia Solis Lemos is an assistant professor and data scientist at the University of Wisconsin-Madison. And finally, Hetty Baxter-Laufer is the Director of Teaching and Learning at the Wisconsin Fast Plants program.
So I'm going to tell you a little bit about the investigation, setting it up so that you're prepared for the second half of the webinar when we'll really go more in depth with the analysis part of the investigation. Some of you might be familiar with the Fast Plants selection investigation that comes out of the AP Biology material and also has been on our website for a long time. Our program out of the University of Wisconsin-Madison has been around for over 30 years. And way back in the day, we started noticing that the number of hairs or trichomes around the margin of leaves is a trait that could be selected for and increased in future populations. And so we wrote up a lab that has gone through various iterations, but has been a really successful way to teach selection hands-on, in which students count the trichomes that are around the margin of the first true leaf as they're growing their plants, and then they selectively remove any that are in the bottom 90% of the population for the hair count, and intermate those that had the highest number of hairs and breed for high hair counts, and look at the difference between the parent generation or the first initial generation and the selected generation. And that's been a great process for being able to have students gather firsthand data that they can then manipulate and analyze to look at the effects of selection. And there's certainly a lot of analogy between the artificial selection and natural selection that makes it a real natural, so to speak, for teaching those concepts. But now we have polycots, and the reason why we're leaning towards polycots as a really good alternative is that this seed stock, which we've been working on for about 10 years, it's not a Mendelian trait, but it is a trait that doesn't appear to be influenced by environment, or at least not that we've been able to detect yet. And we see that when we select for polycots and against dicots, we can increase polycot frequency in just one generation. The other thing that's a great advantage of it is, unlike the trichomes or the hairs that are on the first true leaf, those you have to wait, you have to grow plants in a soilless medium and grow them long enough to be able to see the emergence of the first true leaves and then count those hairs. Whereas polycots, the cotyledons emerge as the plants are first germinating, so we get the ability to look at and score seedlings for how many cotyledons they have within about 4 to 5 days. And the other great advantage is that now we have developed some techniques so that we're able to produce at scale generation 1 and generation 2 polycot seedlings. So that makes it possible for us to abbreviate the selection experiment, which has some disadvantages and lots of good advantages as well. And so we're going to talk a little bit about that, polycots as the subject of selection. In case you've forgotten your plant biology a little bit, just a little bit about cotyledons. Cotyledons are also referred to as seed leaves, and they actually develop with the embryo inside the seed, and then they emerge. There's one of the first things that you see emerging after the little initial embryonic stem or hypocotyl. These solar panels that are cotyledons unfurl, and that's how the seedling is able to get the energy it needs to be able to continue to grow and develop. And so in the next slide, we have actual photos so that you can see those cotyledons are very pronounced at a young age. These are polycots grown in soil in the picture on the left, and you can see that the one picture has 3 cotyledons. It's a relatively rare trait, and in some plants, that is a deleterious condition. It's associated with other traits that make it not as successful in reproduction. But we've bred for them for the polycots to be able to reproduce, and it's a very easy to observe trait. Now, for those of you who are a little new to fast plants are a research organism or a model organism that was developed at the University of Wisconsin-Madison for having a really rapid life cycle and the ability to produce seeds that can be planted quite quickly in the process of the development cycle. They don't have a dormant stage. So for an extended investigation into selection, your students can actually intermate their plants, which go to flower in just 2 weeks after they've been planted, if they have the typical standard growing conditions, and produce their own seeds that they can then harvest and grow the second generation seeds. But with the polycots, and because of our experience, we know that another option is to have the seeds just germinated on paper towels. So you can see here in this slide a kind of a comparison of a couple of different options of how this investigation could play out. It could be that you would get the generation 1 polycot seedlings and do something similar to what I described with the hares, where students are breeding their own plants for the second generation. And that is great in terms of getting to know the full life cycle of plants and also really conceptualizing where those generation 2 seeds came from. Or in the case of option B, which we specifically thought about in terms for supporting the idea of distance learning, where students could have seeds at home that they germinated and scored and shared data back to a shared spreadsheet. That option is doable with the polycots, and it's a much shorter time frame needed, and so there's pros and cons with both ways. But for today, we're going to focus in this webinar on option B. And I'd like to turn it over for a little bit at this point to one of our collaborators who's helped in a lot of different investigations, Bob Kuhn, who's coming to us, I think it looks like from the classroom still today. It's already back to school. So I'm going to pass it off to you, Bob.
Hello, everyone. Some of you are probably like me, either already teaching or in pre-planning, trying to get your head around what we're going to do and what kind of labs we can possibly do this year. It's a big challenge, but we're up for it and we'll figure it out. And I think this comes at a great time, especially this option B. This option B that will entail growing the seeds on paper towels and Petri dishes or something similar is great because you can still have your kids make observations. The time is shorter to actually have them be able to collect data and think about it. And then the great thing is that they have 2 generations to compare. So in a period of, let's say, 10 days or 12 days, they'll have had 2 generations that they can look at side by side, that they can examine up close, that they can do qualitative and quantitative analysis, and they can really do some great inquiry. You could do this at home or at your school if you have access, and show it to them on camera or take pictures. You could even potentially give them seeds if you have the ability to do that, and they could do this at home, and then they could collaborate and share the data. So in this slide, you see the basic setup here. So the only materials you really need for this are some paper towel. I think the Fast Plants likes the Bounty paper towels, so that's what I always pick up if I can, but I think any paper towel will do. And cutting out the circles on the smaller sides of the Petri dish. If you don't have a Petri dish, you can use some other sort of deli container. And you wet that and align your seeds, sort of like you see in the center picture there. And then probably the easiest thing to do is to make a well with your Petri dish in another deli container that allows you to wick the moisture up onto the paper towel, and that provides water for the seed. So very low tech. Most of these items would be available to students, or they would be easy to deliver to students. And the next slide, obviously, besides water and something for the seeds to grow on, you also need light. And depending on the situation that you find yourself in, this could be a challenge, it might not be. You could go for something as low tech as a windowsill with sunlight coming in from a good window. You could use a lamp, desk lamp, or a shop lamp, or even if you're in your own classroom, maybe you have grow lights that you're going to grow these plants to show the students. Any light will do, but the thing to remember is that depending on the intensity of your light that you're using, that is going to change the time that the seeds will grow. And so you just want to factor that in. If you use a windowsill, you might not see sprouting at the same rate as you would if you were using a 24-hour grow light. Another thing to think about is once the cotyledons start emerging from the seed, that sometimes you might need to use a toothpick or the end of a pencil or something to sort of get at and pull the cotyledons apart a little bit to count them. But this is what you might see after a few days of growth. Here we have 15 seedlings that are growing on the paper towel method. Notice that they're sitting down in a water well and the wicking up through the paper towel. And so using this as an example, you can see how it would be really easy for students, either through a picture or a webcam or some sort of demonstration, to be able to count. And so we're going to be able to get quantitative data from this by counting the cotyledons. So let's do this. I don't have access, I don't think, to the chat or the questions, but if you could post what you think on this, see how many polycots you can spot out of these 15. So a polycot would be 3 or 4 seed leaves and not 2. And I'll give you a hint. If I'm not mistaken, the one in the upper left is a dicot. Okay, so that's a top seed leaf and a bottom seed leaf there. And so we'll pause here for a second. Let's see if we can maybe get in the chat some input of how many polycots. That's either 3s or 4s that we see.
Yeah, if you'll just type your answers into the questions, I'll start to kind of read them off. So we have somebody who says that he thinks that he sees 6. Someone else sees 5, 8, 6. So I think this does illustrate a little bit some of the challenge of doing this by image. You could do it this way, but we are getting a fair amount of variation, and it really is a lot clearer if you can sort of move them around to see them.
Yeah, so if you have the chance to look at them, you know, with a magnifying glass, or even increasing the size of the picture to be able to see them up close, it helps a lot. I thought there were 7, but I'm not a fast plant expert, so maybe Hetty will tell us the real answer soon. So I guess in conclusion, before I hand this back, you know, you really have an opportunity to use these polycots to design an inquiry lab with living things that students can either grow on their own or they can watch you grow. They have 2 generations, and they can do a lot of comparison. I'm planning on using this in my AP biology class this year. I will probably grow the plants and share pictures, just the situation that I'm in. But if that changes and I'm allowed to give them seeds to grow, by all means, I would rather them be doing the exploration. So I'm going to hand it back to Hetty at this point, and she's going to talk to you a little bit further.
Thanks, Bob. And I would just reiterate that if you are able to pull the seedlings apart a little bit, it takes some practice to count the, to recognize a polycot from a dicot. It's quite simple though, especially if you destroy the little seedling and pull apart its polycots or poke around and are able to look at it firsthand. And since we're not going to be growing these beyond the seedling stage, there's no harm in pulling the cotyledons off and actually looking at them as individual cotyledons. And if you can't, like in Bob's case, if you can't send seeds home with students to do that, you are able to either take pictures or use a webcam, you could do that with every seedling and put the cotyledons near them so that you could tell the dicots from the polycots more clearly, if that felt like that would be helpful. Again, because we're focusing on this distance learning of the option B, we're not actually growing those plants in soil, and we're not going to actually implement the experimental design that students would come up with for how you test to see if you could increase the frequency of polycots in the second generation. However, it's really important that students do that step, and that's our chance to check for their understanding that these seedlings that they were looking at in a Petri dish could be part of an experiment where they would be grown to flower and reproduce seed that we could then germinate and look at to compare the first and second generations frequency of polycots. And so in the next slide, we have a kind of an idealized example of a way that you could do this remotely. You could have students put together a slide deck in Google Slides or on PowerPoint, or turn in a shared document where they would describe how that experiment would need to unfold. And that would take understanding variation, understanding that the trait that you're investigating needs to be heritable, all those things that we need to understand to really grasp the idea of natural selection. So you'd want to make sure that there's a check-in for understanding that experimental design. And because we're doing this truncated version, everyone's going to be looking at this one trait, polycots, and trying to increase it. Whereas if it was a more open-ended investigation with polycots, you could try and select against the polycot trait, or you could look at these plants and select for something else too. But we're focusing on increasing the polycots. So once the experimental design is completed as a reasoning exercise, then it's time for students to get seeds to germinate and gather data from a generation 2. So we're kind of pulling a Julia Child. Now we have the generation 2 seeds without having had to produce them. And
I'm going to break in just for just a moment because we've got a little bit of confusion in the questions. So we actually are able to provide both generation 1 and generation 2 seeds that have been already produced for this type of investigation. If you're familiar with our monohybrid or dihybrid genetics kits where there are multiple generations provided in the same kit, it's the same kind of principle. So I just wanted to let you know that had come up in the questions, and I wanted to make sure everybody was on the same page.
Yeah, great clarification. Thank you. Yes, and that's one of the reasons why the Polycot seed line is being released at this time, was it takes time to figure out how to do these things to produce that second generation seed and consistently have a range of polycots that will show up in the seedlings that's like what you would have with a generation 1, and then it really is a generation 2 set of seeds but we've produced them for you, so that again it gives this option of being able to pull a little faster timeline. You could even use those generation 2 seeds if you had students produced their own seeds in generation 1, but you didn't want to wait for them to get fully mature the first year. You could use our generation seeds. So anyway, it gives us more options to be able to offer both generation 1 and generation 2 seeds. So thank you, Julie. So now we have the generation 2 seeds, again, germinated on a paper towel in a, in the setup similar to what we did with generation 1. And students can collect the data on generation 2 and compare that to what they had in generation 1. And so where we are in the investigation is at the point where it's time to analyze data and really understand how we use those data that we can collect from the seeds and direct observation as evidence for an explanation about whether the selection experiment worked or not, or whether we were able to cause a change in the second generation. And to look at that data analysis, I'm going to hand it off to my colleague Claudia, who is a really skilled data analysis scientist, and it will help us with some conceptualizing the processes and how you can help students understand this too.